Okay, I think we'll go ahead and get started. So I'd like to welcome everyone to the first Cancer Center seminar of the academic year. It's my honor to introduce Dr. Lynn Matrisian. So she earned bachelor's and PhD degrees at the University of Arizona before doing a postdoctoral fellowship with Pierre Chambon in France. She then went on to become a faculty member at Vanderbilt University. So she was there for 25 years as an R01 funded investigator, rising to the level of the chair of the Department of Cancer Biology. And her work there focused on mechanisms of cancer metastasis, cancer cell stromal interactions with a particular emphasis on matrix metalloproteinases, and is well known for her work in this area. In fact, she was the first to clone one of these MMP genes. She published over 400 papers. She is the former president of the American Association for Cancer Research, or AACR, and became intimately involved in National Cancer Institute priority setting and strategic planning. And then most recently, she's moved on to become the chief science officer of the Pancreatic Cancer Action Network. And so she's here to share her many perspectives in cancer research and oncology. And thank you for being here.
Well, thanks. It's an honor to be, the first kickoff speaker for your seminar series for this year. And I'm going to start by with some statistics, and statistics that we all know, but every time I see them, I get a little bit of a jolt. And I'm going to tell you a personal story. So I'm going to tell you about my friend Holly. And Holly and I grew up together. We were next-door neighbors. We were born 6 months apart, first children, and so our mothers learned how to be mothers. And we went to school together. We went all through grade school and junior high school, and went to the same high school. And then we went to different colleges. And Holly was always very athletic, and she was involved in all sorts of college sports, but she was having trouble with her leg. And so she came home for Christmas one year, and her mother took her to the hospital, or took her to the doctor, and they put her in the hospital right away. And she was diagnosed with malignant sarcoma. So Holly graduated from college, but she died shortly thereafter. So that was right at a time when I was deciding what I was going to do with my life. And I was working at Hershey Medical Center. I was a medical technologist working in the hospital laboratory. But the person I worked for also had a research laboratory. And I said, ooh, this is really kind of cool. And I think I need to do something about cancer. This isn't right that this can happen to a 22-year-old. So I loved biology. I really thought biochemistry was really kind of cool, but molecular biology was the brand new thing at that point. This is in the mid-'70s. And so to marry, in essence, molecular biology with cancer biology seemed like where I should go. So I'm going to tell you a story from my perspective, my many perspectives on cancer through these 40-some years that I've been doing this. And I'm going to use the blind men and the elephant as the framework for this, because the blind men are actually very much like me and like many of you, in that they are to learning much inclined. They are educated people who were very curious, that they wanted to satisfy their mind. They wanted to touch the elephant and see what this was all about. So my first kind of the basis is the elephant's body, the wall. And then for me, that's being a molecular cancer biologist. So as a postdoctoral fellow, I went to a laboratory that was very into molecular biology techniques, but didn't really care what they applied it to. So I said, well, what I want to know is what's downstream of that one gene, polyoma middle T, that transforms a rat fibroblast from being perfectly benign into being able to form a tumor and kill a mouse. So we did subtractive hybridization, and we came up with two genes. One of them was only in the cell line that we had been looking for, but the other one was broader. And so we actually sent somebody, we had to fly them to England to look at the database because there was no databases online to see what the sequence was. It was novel. So we called it transin, or transformation-induced gene. So then we had to figure out what it did. And through a whole series of experiments, we ended up realizing it was a secreted protein that had proteolytic activity. Now, at first, I was rather distressed because I said, I want a growth factor. What's this protease? It's just something you put inhibitors in your solutions all the time. What good is a protease? But then, of course, there's this hypothesis that if you really want to study cancer, the thing about cancer that makes it cancer is its ability to metastasize. And that ability to metastasize involves proteolytic activity. So then I said, okay, this is something that I want to pursue. So I used that as the start of my laboratory. And it turned out that transin was stromolysin, or MMP3, so the first member of this whole family of matrix metalloproteases to be cloned. So that was a very exciting time. We were cloning other members of the family, and we were looking for their biological activity. And as a molecular cancer biologist, I wanted to know, how do these things work? What is their substrates? What are they doing to the biology of the tumor? So we did a lot of experiments. We worked first in skin cancer, and then colon cancer, and then breast cancer, and looked at individual family members, looked at what their substrates were, looked at how they were involved in the process of tumor progression. And so every time we looked, it was, if you had MMPs, it made it worse. If you inhibited MMPs, it made it better. So this turned out to be quite exciting. So that led us into the second perspective, was the pharmaceutical company said, aha, now this is a very interesting approach. We can inhibit metastasis if we can inhibit these enzymes. So that brought us into this whole sphere. People were asking for my cDNA, wanting to express the protein, crystallize the enzyme, find small molecule inhibitors that would inhibit the activity of MMPs. So it turned out that whole field exploded. A number of pharmaceutical companies got involved. There were a dozen large phase 3 clinical trials using MMP inhibitors that, in essence, crashed and burned. So a lot of these studies showed no effect, but some of them showed that it actually, at interim analysis, there were more people. They had a worse prognosis, a worse survival on the experimental arm compared to the control arm. So it stopped all development, all drug development with MMP inhibitors in that company and eventually in all companies. So because it was certainly much more complex than our mice had told us. So what went wrong? Well, there's a lot of things that could go wrong, and the answer is virtually all of them went wrong. And there's a little bit of perhaps a reason, but it was actually only 7 years between the time that the first cDNA clone, where we saw the sequence, and treating the first patient with an inhibitor. And this is the late 80s, early 90s, so that's incredibly fast at that point in time, and we really didn't know much at all about that whole process. And yet there's still a lot of things that we could have done better. So that brought me into really the realm of translational cancer research. And I started originally in Vanderbilt. The basic scientists were on one side of campus, and the clinicians were on the other side of campus. And I said, no, we're going to have a Department of Cancer Biology. We're going to embed ourselves in with the clinicians, and we're going to understand this better, because we put all this effort into these studies, and look what happened. There was just a wall between the basic scientists and what happened in the clinic, and we have to understand that better. So that really led to trying to understand, the way clinical trials worked and what was needed in order to get up to that point. So now we could start to look, and the truth is that I had early on had some mouse experiments that said that you don't want to inhibit MMPs in these experiments. And I had tried to publish that, but everybody said, oh, no, that can't be right. You must have done something wrong. Go do more experiments. Go do more. I could never publish that until the clinical trials failed. And then you could go back and say, okay, now you can start publishing studies with different MMPs that actually show that they're anti-targets and you don't want to inhibit it. So we started to uncover the complexity of this family. There are different family members. They do different things they have different substrates. They work in different cell types. They work differently in different tumor types. So really starting to unravel that and being, in essence, much more honest about what we're actually finding in these experiments. So one of the things that we just I'd never even thought about when we first did these experiments was I need to know whether the drug is actually hitting the target and modulating anything. Because if it doesn't work, did the drug not work or did the hypothesis fail? It doesn't matter if you inhibit this activity or not. So once the clinical trials failed, my lab had to kind of reshuffle and figure out what was important now. And one of the things that was important was coming up with a biological indicator of whether an MMP inhibitor was inhibiting MMP activity or not. So we spent a lot of time on molecular imaging using near-infrared probes so that we got in vivo sensitivity. It's a probe where it has a peptide that's cleaved by different MMPs, and we can dial in specificity for different MMP family members depending on that peptide sequence. And it has a reference on it, so you can look at sensor reference ratio and get some sort of quantitation as to the activity of a certain MMP in vivo in a tumor or in an inflamed site. And then we could show that when we treated these mice with MMP inhibitors, we could, in essence, modulate the activity that we were reading out from these probes. And we could never get more than about 50% inhibition with the inhibitors that were available. So whether that's not enough, it's a tool that we now realize that in any drug development you ought to have a pharmacodynamic marker so you have some idea of whether the drug is hitting the target or not. So that brought me kind of that experience in saying I have to understand. And I think there's nothing more than failure to make you really dig into something. When something works, it works because you knew it was going to work. It works and you just kind of keep on going. But when it doesn't work, you really have to think about what is the biology behind this, what is the processes. So I had been thinking very hard about what went wrong and how we, as a basic scientist, I need to understand better translational research. So at that time, the head of the NCI, Andy von Eschenbach at the time, had set up what's called the Clinical Trials Working Group. And once that got started, he said, I need one for translational research as well. So he asked me and two others to lead a extramural committee. We had 63 members of this committee to take a look at translational cancer research and say where are we, what needs to be done, how can the NCI help with this process. So we worked for 2 years, we got this report, and at that point then Ernie Hawk, who had been at the NCI for a long time, decided to go to MD Anderson. And so there was no one to lead the project from inside the NCI. So I had the opportunity to work half-time. I would work 1 week in Bethesda and 1 week on my laboratory at Vanderbilt. But I gave up all my administrative responsibilities for those 2 years so that I could, in essence, do this project. And we really focused on what's called type 1 translation, so translational research from discovery up to early phase clinical trials. And we decided we had to operationalize this, that we needed a flow chart. How do you do translational research? So we had a definition, but it doesn't really tell you what to do. So we used engineering diagrams. So diamonds are decisions, boxes are activities, and arrows tell you which way to go. And we decided that the goal, so what you really want is a goal. The goal we could define as one of 6 different clinical goals. So 2 were assessments, so either biomarkers or imaging. And 4 were interventions, so drugs, immune response modifiers. The immunologists say it's not the same as a drug. It's a different group. You need a separate pathway for immunotherapies. Devices, so things like radiation and surgery, and then even lifestyle alterations. So those are clinical goals that you're trying to accomplish in translational research. And so we could map out pathways for all of those clinical goals. And each of those pathways have different components. One is credentialing, which is where we spend a lot of time as kind of basic laboratory-based research. So is this scientifically valid? And then recognizing if there's a clinical need and if it's feasible or not. So that's the kind of work, but all that does is get you through the diamonds and tell you whether it's worth really doing the translational research that's required to turn that into a good idea. It makes you know it's a good idea, but to turn that into something that you can then actually accomplish, get it into early phase clinical trials. So through that, you have to create the modality, so the drug, the imaging agent. You have a lot of supporting tools that are required along the way. And then you have to develop it in terms of the toxicity. You have to test it before you can go into early phase clinical trials. So each of these pathways, in essence, has been developed. They're published. They're available if anybody's interested. And you can use these for a variety of things. One, simply to understand, if you're credentialing something, what's going to come next? What do I have to think about if this works the way I want it to work? And we all think and hope and work very hard at making our projects work. What's going to come next? Let me be prepared. So that you don't end up like MMP inhibitors, where we, in essence, got totally derailed by not understanding what was going to happen in the clinical setting. So we then had the opportunity to create projects. So how do you use these tools then? And so how do you get across that valley of death in order to get from a basic science idea into the clinic? And everybody had their ideas. Everybody said, well, we need more of this. We need more of that. This is the problem. Everybody has a different problem. And every time we looked, the NCI had a program that would address that problem. Yes, but there's not enough money in those programs, of course. Well, the bottom line is there's not enough money to fill that gap. What we were saying was, if you want to walk across that bridge, you have to make everything come up to the top so that you can just walk across. There's not enough money in the world to do that. So how are we actually going to do a translational research project? So what we decided is what we have to do is decide that a project is right. It's gone through credentialing. It's ready. This is something that's needed. It's feasible. It's scientifically valid. Then you want to leverage existing resources. And again, this one from an NCI perspective, but it's true in cancer centers. It's true in lots of places. What existing capabilities are there that can be leveraged in order to do each of those boxes that are in that pathway? So then you have to add additional money. You have to be able to get from that, in essence, existing platform into your project, using the pathways as a guide. And then you need project management. You have to have somebody with business sense, with operational abilities that can help get you into each of these, get you through the pathway, get all the stars aligned so that you can go from the top to the bottom into clinical trials. So that was the concept. We had the opportunity to test this at the NCI with a couple pilot projects, and it seemed to be like it was working pretty well. However, then my 2 years was up, the director changed, all sorts of things happened, and in essence, nothing's happened since that time. It's all kind of sitting on the shelf. Well, I was not very happy about that because I believe in this. I said, okay, there was a lot of smart people who put their minds together, who had a lot of ability, and it makes sense to me that this is a way to go forward. So I decided that, I wasn't going to live long enough to see the government do this as a bottom line. And so if I wanted to be part of it, I had to kind of change my emphasis. I had to do something about it. So, kind of along the way, I decided that it's philanthropy, it's the advocacy perspective. Here's a group that is used to doing things fast, that has, doesn't have all the barriers that the government does. They have to be absolutely fair. They have a lot of processes, a way to kind of accelerate this whole process and perhaps do it in my lifetime. So I actually went to business school because I knew nothing about business, and it wasn't actually necessary that I did that. But as an academic, what do you do when you don't know something, right? You go to school, so that's what I did. But along the way, then, I got this opportunity to interview at the Pancreatic Cancer Action Network, and they were the only organization that had come to see me when I was at the NCI to say, what are you doing? How can we help? This is really interesting. And so I interviewed and I fell in love. And now I have been for almost 6 years now at the Pancreatic Cancer Action Network. It was a hard transition. It was certainly not easy. I had spent my whole life in academics, and I loved it, and I loved my lab, and I loved my department. But I just felt that for me, this was what I needed to do. So the interesting thing about being in an advocacy group is that you're pretty single-minded. You have a very specific goal. And the thing that I liked in particular about the Pancreatic Cancer Action Network was that they had more than one tool in their toolbox to accomplish that goal. And so we use this wage hope. This is our rallying cry. And we use the 4 corners to indicate 4 different approaches to waging hope for pancreatic cancer patients. So our goal, we can define it. We use measurable goals. We started this in 2011 when we had 9 years to double survival by 2020. Every morning I get up and say, how many days do we have left until it's 2020? I got to get moving, right? But it gives you that real sense of urgency, that real direction, and every decision we make is based on, is this going to help us get to 2020? Because if it doesn't, you don't do it. Okay, so it makes decision making a little bit clearer. So we approach this through 4 different ways. One way is through government advocacy. So I work in the DC office, so the other people in the office are mostly policy people. I mean, they know how the government works. And so we have been able to influence that as an advocacy organization. We've been able to advocate for NCI funding. We look at the NCI funding for pancreatic cancer every year, and it continues to go up. The numbers, again, the government numbers are only current as to 2014, but as of that year, there's been nice increases. And part of this is because of our advocacy. We don't give up, okay? So we bring 500 people to Washington, D.C. one day in June every year, and we storm the Hill with purple, and we say, you have to do something about the NIH budget. And we said, you also have to pass something called the Recalcitrant Cancer Research Act. So we work behind the scenes with legislators in order to get them to pass this bill. And it took us 5 years, a lot of emails, a lot of phone calls, a lot of visits to the Hill, but we got it passed. It was signed into law January 2nd, 2013 by President Obama. And what it says is the NCI needs a strategic plan. That's all it says. There's no money in it. That all got stripped out because it wasn't going to get passed if it had any money in it. But it's developed a scientific framework, which is a strategic plan for pancreatic cancer. And what happened then, they called a special group together. They picked 4 priorities, And at this point, they have funded more than $63 million, and some of these are still ongoing and still more money is being added to it, of research in pancreatic cancer that would not have existed had we not gotten our advocates involved, gone to the Hill, and make that kind of difference. So it's a way to impact the field at this broader level. In addition, we have our own grants program, so we raise money for grants. We have funded over $40 billion at this point since 2003 to 158 scientists in 58 different institutions. And we're so delighted now to have a grantee from University of Minnesota, so Dr. Stroms, who is just new here, wave. And we're delighted to have her join. In essence, we do consider this a family. We have meetings every year. We try to encourage collaborations between scientists to, in essence, build the field. And we think some of our initial investment in early career scientists has been part of what's resulted in the increase in funding that we've seen, because people are writing R01s, they have pilot projects, they have preliminary data, they then get funded. So it's kind of part of something that we've worked on for a very long time, but we think we've contributed to building the field. So that's government advocacy, our own research program, patient central, one-to-one patient support. So we have a call center. We have a dozen full-time employees that answer the phone, answer emails on a daily basis, that give one-on-one patient support, disease and treatment information. We have a clinical trial database that we keep up to date. So clinicaltrials.gov is updated occasionally. This is updated on a monthly basis. We know what sites are open. We know whether patients we should send people there or not. Specialized information, support resources, all sorts of information. And it's a case management system so that if a patient calls in, 911, I just got diagnosed, what do I do? 3 months later, I'm on treatment, now I'm having dietary problems, what now, what do I do? They go back to the same person, they get information that's really relevant to their particular situation at that particular time. So we've served over 165,000 contacts, so different times that we've talked to somebody and helped them, and over 33,000 in a year. So it's a very robust program to help patients on an individual basis. Our clinical trial finder is available. It is open to patients to do searches, or to healthcare professionals with different sets of filters. If you're interested in knowing what the current landscape looks like for pancreatic cancer clinical trials, you can let us know. We'll give you an account, go in and search the database as much as you want. And the goal here, of course, is to increase enrollment into clinical trials. So because we have this database, we have relationships with all trial sponsors. Every 3 years, we determine how many patients went on pancreatic cancer clinical trials in the United States. In 2011, it was 3.8%. In 2014, it was 4.2%. And for those people who call Patient Central, who get a clinical trial search, our estimate is it looks like for the people who respond to the survey, 12% of them go on to clinical trials. So we're trying to continue to move that needle by getting people onto clinical trials. And then, of course, the fourth thing is raising awareness and across the United States. So we have 60 affiliate networks across the United States. They raise awareness, they raise funds, they provide about 1/3 to almost 1/2 of our revenue each year. And the Minnesota group is very active and very involved, and their Purple Ride Stride is this coming weekend in Maple Grove. So just a shameless advertisement for our affiliate network. We have 140 employees at the Pancreatic Cancer Action Network, and a good deal of them manage the volunteers and help with these kinds of activities. And Scott Nelson is on our Survivor Council. He helps advise us on the activities we should do from a survivor's perspective. and has been very involved and out to our headquarters many times at this point, several times at this point, to help us. So we really try to, again, do as many different things, try to, and I think, again, that's one thing, is having those four corners really gives us an advantage, a ability to leverage those different ways of influencing the community. Okay, so now I'm at the Pancreatic Cancer Action Network, and now I have to think broader. I have to think more like a population scientist, right? So I have to think about kind of landscapes and trends and those types of things. So we looked not too long ago at, there was a paper that came out that looked at demographic changes and what that was going to do to cancer incidence and cancer deaths out to 2030. But that didn't incorporate individual cancer types. So all I did was break it down and I used some of my MBA statistics in order to look at, a percent increase per year and how you kind of leverage that all in it in order to project that pancreatic cancer, in terms of the number of deaths, is going to surpass breast cancer, which I predicted, and it did this year. There's now more deaths from pancreatic cancer than breast cancer, and it will surpass colon cancer in another couple years. So it will be second only to lung cancer by 2030 and probably by 2020. So that was, again, a whole different way of thinking about cancer from a population-based perspective. We look at clinical trials. We look at trends in clinical trials. We noticed a number of years ago that there were all these trials for neoadjuvant treatment for patients who could go to resection. But there's not that many patients that can go to resection. So those trials were never going to accrue. Whereas there were very few trials for patients who had already been treated, who had metastatic disease but are looking for recurrent treatments. Those patients really want clinical trials because they're out of options. So this whole system wasn't set up with thinking about what patients need. They were thinking about what kind of trials people wanted to design. So part of our job is to publish papers that look at these kinds of trends, bring some sort of awareness as to these kinds of trends to the field. We looked at, we have two standard of cares, in essence, for pancreatic cancer in recent years. So that makes three total. Gemcitabine plus Abraxane, and FOLFIRINOX that we use for metastatic disease. So, but there's been lots of phase 3 clinical trials. So how come so many of them failed? So we did a deep analysis on looking at those that had phase 2 results and that then went to phase 3. So why did we get fooled? Why did the phase 2 data say we should go to phase 3 when the phase 3 failed? And what kind of benchmark should we set so that we have fewer failures as we go forward? How can we better use that phase 2 data? And again, publish paper on what we think those benchmarks should be, and based on historical data, whether those are predictive or not. So again, a much different way to look at cancer from that particular perspective. And the important thing is, if you go back, had these benchmarks been used, there were over 11,000 patients that went on to phase 3 trials that failed. If we could have used them for phase 2s to figure out what was the next, what was the promising new treatments for pancreatic cancer, perhaps we would be much farther. So those kinds of perspectives, those kinds of looking at the field from that much broader perspective. So trying to put it all together is saying, when I got to the Pancreatic Cancer Action Network, we had strong one-on-one patient support. We had strong research. We were supporting research out in universities. But they were two independent things. They were separate corners. And I said, there's opportunity here. We really should be doing things that help individual patients, but we learn from them so we can help future patients. I am a believer in precision medicine. I believe there's information in tumors, the molecular information in tumors, that will help determine better treatments. Our scientific and medical advisors said precision medicine won't work in pancreatic cancer. You can't get the tissue. It's all KRAS driven. There's nothing there that it's not going to help. Okay, let's see if that's true or not. So let's help facilitate getting molecular profiling in the community. We have patients calling us. More patients call the Pancreatic Cancer Action Network than go to any individual site throughout the United States. Let's use that. Let's get molecular profiling information and see whether that's true or not. So we set up something called Know Your Tumor. They call us, patients call us. We determine if they're eligible, very broad eligibility requirements, but you have to be able to get tissue, and you have to have enough time to get the results back before you need to make your next treatment decision. So those are the two main criteria. We then partner with a company called Prothera. Prothera does all the work with talking to the interventional radiologist, figuring out how much tissue. They get the tissue, they send it out. We're agnostic as to where they send it. At the moment, we use Foundation Medicine and neogenomics, so we do some immunohistochemistry, a whole panel of genomics. We do some phosphoproteomics kind of on the side for research-only purposes. They get that information back. They have a lot of bioinformaticians, computational biologists that are setting up algorithms to figure out what you do with that molecular information. They then have a medical review panel that looks not only at the molecular information, but at the patient. What have they been treated with? What can they be treated with? And then we generate a list of clinical trials, off-label treatments, or standard treatments that are options for that patient, and those options are ranked. Again, this is not medical recommendations, but it's information that the treating physician can use to treat that physician. And then, of course, we keep all that information. And the patient started with us. We can talk to the patient. We can call them back. We can say, have you made a decision yet? Can we help you make a decision? Did you know that such and such a resource exists? So we're able to help with that. And then we have a patient-reported registry, so patients can also, whether they can go through Know Your Tumor or not, tell us all about their experience, tell us about their symptoms, tell us how they've managed that, and we can mine that for things like we have a couple abstracts in now, poster presentations for taking digestive enzymes and how you take it and whether it works or not, and all kind of the experience of patients throughout the United States on that. So Know Your Tumor is now in its third year. We have 680 reports back as of the end of July. We are surprisingly quite successful in getting first-time 91% of the time we get molecular information on the first try. We can get that up to 99% if you're willing to try again or do some other activities in order to be successful. A third of them come from community physicians. The rest come from what we would consider high-volume centers, either academic institutions or high-volume specialty clinics within the community. And we've hit 48 states, so that includes Alaska and Hawaii. We have, I think, Rhode Island and Delaware yet that I have to work on in order to get them on board. And we have, anecdotal evidence that you can find molecular alterations and you can help patients. So patients who have had, get on a melanoma treatment because they have BRAF V600E mutation and, is doing very well in response to that. So if we look at the data in general, that's interesting. Okay. The data is 27% have highly actionable alterations. 50% have nothing that nothing that we can particularly do anything with. The rest have things that are like in pathways, and maybe it's interesting, but not really. And that's a whole list. Sorry about that. Of the kind of alterations we see. Certainly BRCA1, BRCA2 on PARP inhibitors. Things like ROS and ALK fusions and those kinds of things in 1% indications that have molecular, that have targeted therapies that we can put patients on. The outcomes, so we get outcomes from these patients. We call them back. Prothera calls them back, bugs the doctor. We get outcomes from the patient. We collect all that, and again, it's 21% of patients who go through Know Your Tumor go on a clinical trial. So we're up to, at least in that population, 21%. Some of them go on off-label targeted treatments. And if you look at, again, the statistics on progression-free survival of those that have a highly actionable alteration and they go on matched therapy, that their progression-free survival is longer than those that have no highly actionable alteration that, in essence, go on standard of care. The numbers are too small for those that have something that's highly actionable and they don't go on something that's targeted to it, so that happens, but luckily not all that much. So the comparison is with those that have no highly actionable alterations. So we're preparing this for publication, but I think it says that, yes, you don't have to ignore pancreatic cancer when it comes to trying to take advantage of the targeted drugs we've developed in other cancer types. So that brings us then, we're dependent through Know Your Tumor on the clinical trials that are out there that other people are doing. And we decided, okay, we need to kind of do our own clinical trials. We understand the clinical trial landscape. We've done all these studies to understand what's happening. Can we support a clinical trial network, develop our own clinical trials in order to try to advance this field faster? So that's what we're doing. We call it Precision Promise. It has three components. Of course, the kind of the leadership component of lots of steering committees, lots of people involved. We have 12 clinical trial sites that will start in this consortium. We hope to go farther at some point, but we got to walk before we run. And then we integrate some of our research grants so that we can tee things up and get a pipeline of things that can go into this clinical trial network. So these are the 12 sites scattered across the United States. And our first patient should be able to, we're aiming for like early spring of 2018 to get the first patient in. So we've hired a chief medical officer. So I now have a counterpart who's an MD who's run lots of clinical trials in pancreatic cancer. And so we can kind of really try to so again, 2020 is coming. We're trying to get this going as fast as we can to really start to make these kinds of differences, and then we'll have to set our next goal pretty soon. So Know Your Tumor and Precision Promise are complementary. They really address different needs. Know Your Tumor is for kind of anybody in the community. You have to go to one of these 12 sites to do Precision Promise. We can do newly diagnosed patients in Precision Promise. That we really can't do that in Know Your Tumor. It's you can't get the results back soon enough. And in essence, we're going out talking to pharmaceutical companies, getting the arms. It's an adaptive trial. We're starting with an adaptive trial design, one where we can just put arms in and out, always with a comparison, with a standard of care comparison, but randomized at only 20% of the patients that go onto standard of care. And the results will be FDA approvable. We talk to the FDA all the time, so you can actually approve a drug through this particular process, or you can fail fast, and we'll learn quite fast whether it works or not. And then the whole point is to gather lots of data. And make it available to the scientific community to help us really analyze it, help to figure out what are we learning from it, how do we go forward from this particular point in time. So I think, again, if I come back to the blind men and the elephant, it's very much like the cancer research field, right? I mean, we've disputed long and loud, right? I mean, we get so passionate about our particular part of the elephant, right? This is what it is. This is what's really going to make a difference. And the bottom line is we're all partly in the right and we're all in the wrong, right? Because we're only seeing cancer is incredibly complex. And it's taken me 40 years, right, to kind of walk around the elephant and see all the different parts. But that's ridiculous. I mean, the bottom line is we're not blind. We can open our eyes and we can see the whole elephant. And all it really takes is team science. We simply need to work with other people who come from these different perspectives. And that's the only way we're really going to make a difference on a disease that's as complex as any kind of cancer. But from our perspective, in particular pancreatic cancer, it is what we call the toughest of the cancers. So I think that my message is that there's advantages to looking at the elephant by its individual components. We have to understand it deeply and thoroughly. But to get the whole picture, it's going to take the kind of collaborations and the time of team sciences that we need to really advance this field at the rate that I know we can do. We've got the capabilities. It just takes that coordination and that will to actually do it. Because the bottom line is we're really all in it for exactly the same reason. And I am happy to take questions.
Thank you very much. We're going to do something a little different this year. We have some remote sites that we're going to give an opportunity to ask questions first, and then we're going to bring it back to questions in this room. I don't know that we have any today, but we're going to give them a crack. Let me give you this in the meantime. Do you have anybody who wants to ask a question? All right. So back to here.
Can you speak to progress that's being made in early screening?
Yeah. So again, I'll start from my perspective. Our perspective was to really to double survival by 2020, and we purposely picked that survival, not saying what kind of survival or anything. So we started with metastatic disease, and that's why precision promise and all that stuff. But in order to make any really, the next leap in survival, it's going to take early detection. We need to get more patients to surgery because that's the real hope for a much longer-term survival. So the advance that we got very excited about is from Suresh Chari at Mayo Clinic, who's shown that about 1%, about 0.85% of new onset adult diabetics have diabetes as a result of having pancreatic cancer. So it's called type 3 diabetes. And it looks like type 2 diabetes, but it is an early symptom of pancreatic cancer. And about 25% of pancreatic cancer patients were diagnosed with diabetes within the past 3 years. And a lot of others had it but weren't diagnosed with it, right? So here is a population that's screenable. So 1% is you can actually work with a 1% screenable. They did it for lung cancer and 30-year smokers, right? That's about a 1% incidence. So it's a screenable population. So the NCI has, as one of the initiatives that came out of the Recalcitrant Cancer Research Act, they set up something called the CPDPC, the Consortium for the Study of Pancreatitis, Diabetes, and Pancreatic Cancer. And they are starting, based on Suresh Chari's preliminary work, gathering a cohort of 10,000 new-onset adult diabetics, of which 85 to 100 will end up being diagnosed with pancreatic cancer. So they're collecting bloods, they're collecting, samples. It hasn't started yet. We're still in the process. But what we're saying is that the plan then is to collect all those in a freezer 6 years from now, look back and say, what was different between the samples of the patients who died and those who didn't, right? And we're saying, no, that's not right. Let's do something with these patients as we get them. There are 2,000, papers out there that say, I have an early detection method for pancreatic cancer. I can distinguish normal from pancreatic cancer. But very few people have pre-symptomatic samples. That's the real hang-up, is having those samples before somebody gets diagnosed with pancreatic cancer. So we're trying to raise $25 million so that we can put it into this study and that we can use some of those methods, both electronic health record, data mining, and blood biomarkers and that type of thing, and we funded some of that separately so that it runs alongside of getting this cohort together, so that when we start getting those patients, we can select out some. Now, it won't be, it'll be much greater than the 1%. We'll have to screen a lot more people than have it, but I'm hoping that if we do it a long time at the same time, get those people to screening, we'll be able to downstage that pancreatic cancer and be able to get those people to surgery. So that's kind of our vision, is that I think there's finally a population for sporadic pancreatic cancer, that now there's good work being done on family history, people with strong family history and that type of thing. But I think some of the issues are different, and this really gives us a population of sporadic pancreatic cancer that I think we can do something with, and that's my current project, is to get the funding for it and to get all the logistics worked out. It means working with the NCI, which has some problems but has also amazing advantages. So that's where we're at.
Jim?
Terrific. Thank you. Okay. So the Know Your Tumor program is very exciting. And the question is, what are the challenges for funding the molecular characterization of a patient's tumors, and then driving that, and then bringing that into the list that you talked about in terms of options? It seems like that would be a very expensive proposition. And does Pancreatic Cancer Network fund that, or are there other sources as well?
Yeah, so we fund Prothera, and yes, it's expensive. It's part of my research budget that goes into that, and thus far we haven't gotten to the point where we have more need than we have money. So I'm hopefully that will continue. And so that funds all the logistics of it, makes it possible. The actual tissue gets sent to the diagnostic company, and the analysis gets billed to insurance. So that we have not had problems, believe it or not, we have not had problems with getting the molecular diagnostics paid for by insurance. Because, again, there's enough evidence now in other diseases, and thus far we, maybe I shouldn't say it too loud, but thus far we have not had problems getting that supported by insurance. The accurate analysis. Now all the rest, everything else yes, right, yeah. So that's good.
So I'm also very interested in the Know Your Tumor and the survival data where people were able to get the matched therapy versus those that couldn't. Do you know from that data set, are those people that are responsive to the BRAF as an example, or the people that are finding other matched therapies, are those pancreatic cancer patients who are maybe the 10% that don't have KRAS mutations and sort of the hallmark P53, P16, maybe DPC4? Or are those patients who sort of in addition to those driving mutations have additional mutations that may be targetable? I don't know if you guys have parsed the data out.
Yeah,
like that,
yeah,
I
it's
wonder
all
about the
there.
heterogeneity.
It's all there. I think you're right. I think a lot of them do not have KRAS. I think a lot of them have the BRAF. They have ROS and ALK and fusions and that type of thing. That the paper submitted, it's actually submitted again. But anyways, you know how that goes. But yeah, I have all that data, and we can certainly take a look at it. It's interesting. There's it's about 5%, I guess, of patients who are truly KRAS wild type. And a lot of them do have things like fusions, and they also tend to be those that are a little bit younger. So there's some interesting data there. Yeah.
Yeah, and I think it could perhaps speak to some of the oncogene addiction work and things like that. I mean, you could have a KRAS mutation, but perhaps still respond to another molecular therapy for a second mutation. That's very interesting.
Yeah.
So.
Yeah.
Thanks a lot. So that was a very inspirational talk. And I work with cancer for 17 years by now, and the more in this field, the more I understand that yet the biggest challenge is the clinical translation of our new therapeutics. And I mean all this enormous amount of time, work, efforts, and money to bring your new drugs, your new therapy to the phase 1 clinical trial. And it's especially relevant to us who designed this kind of revolutionary approaches to treat cancer, gene therapy, etc. And all these financial problems, for example, the toxicology studies and the funds to cover the GMP production, it's so costly, yet we cannot rely on the governmental funding. Is there any hope that we will have more help from the NIH to cover the clinical translational studies?
Well, I would never give up hope.
Yeah, it's why we're still here. But sometimes I do feel myself as that blind man on that elephant. So, and what's next?
Yeah. No, I mean, you're absolutely right. I think the NCI missed a big opportunity to be able to figure out how to get some of this done. Now, they do have the SPORE program, and that's what they consider their translational program. And fabulous things have come out of the SPORE program, but they still don't really take one big problem and try to get everybody aligned to fix that problem. And I still think that's, in my mind, kind of the key thing that would really make a difference in this, in overcoming a lot of the barriers that you're articulating. So, I mean, again, I have control over our grants program. So we have set up $1 million and $2 million grants that take a problem and will fund GMP and will fund all those things that nobody will fund, that type of thing, and tries to get people to come together. You get the different expertise that you need and trying to do it. But again, it's a small piece of, what's needed. But again, remember, we're never going to fill that chasm to the brim. We're always going to have to cherry pick what it is and then try to leverage existing things in order to get where we want to go. So I don't have a good answer for you other than it's something that I personally am going to continue to push for with however I can do that in order to get over those barriers, because you're absolutely right. That's what needs to happen.
Yes. Also, I'm developing oncolytic viruses for pancreatic cancer more than 15 years, and I'm also actually the reviewer of PanCans, the early detection group.
Thank you.
But I'm asking a kind of contradicting question. So actually, my father is 3-year survivor for PDAC, and the father of my best friend is 2-year survivor. Both of them, interestingly, found actually in Japan, but found with conventional detection method. One was with ultrasound. The other one was, because he had a history of, male breast cancer. Then actually, because that he's been, analyzed for the CS19-9. So that those conventional things sometimes find early tumor.
Yep.
So then, how we can intervene, family medicine side or population follow-up side? Currently, the problem is 70% of the patient comes up from sporadic cases.
Yeah.
And if they have a heavy family history, they might be followed up.
Screened.
But others not.
Yep.
How we can, pick up those newly diagnosed family or newly diagnosed case from the population, so that how we can intervene, family medicine or community intervention side.
So, I mean, I can tell you what we're doing. I don't know that that's the answer, but one is awareness, so awareness campaigns so that when somebody presents with some of these other symptoms and it's the last thing that they, I think it's, you do the easy things first, right? I mean, it's indigestion, it's whatever, right? But in essence, bringing pancreatic cancer to the forefront as a possibility of that. So that's an awareness issue, and that's something we have a marketing team working on. There is, we are one of the leaders of something called the World Pancreatic Cancer Coalition. So there are 23 countries that have advocacy groups that are getting together, and that's going to be their thing. There's World Pancreatic Cancer Day in November 17th or something like that. You know, again, this awareness thing. The second thing is the new onset diabetes cohort as a way, and we're starting with CA19-9. I mean, we're screening patients for weight loss and CA19-9, simple stuff, right? To say, and then they have new onset diabetes. So that those are simple things. Yep. 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 Yeah. No, it is. I mean, and that's, I guess, one thing a role that an advocacy group can play is that we can pull on, you know, we have a lot of social media. Somebody who told me that they saw a tweet from us this morning about things. So we do a lot of social media, getting things out. We don't have the budget for national TV ads. I mean, we're just not going to spend, I'd rather spend that on research than national TV ads. So we have to figure out other ways of doing that. So but I think, again, that's our responsibility. You're exactly right to try to do that, because I can pull on more than one string, right? We can do through the advocates, we can do through research, we can do through patient knowledge, so it's all these different ways.
And again, copyright is very important because local news, we pick up those kind of stuff, and this kind of activity, local activity, is really important.
Yes. Yep.
Well, thanks so much for sharing your experiences. So let's thank Dr. Matrisian one more time.